I'd like to invite our first speaker, Todd Vanderah. As I said before, he is the head of the Department of Pharmacology at the College of Medicine. And when I started investigating this area of research, I was amazed to find out that many, many years ago, there was another person at the university, his name is Paul Consrow, who did extensive set of investigations with cannabinoids. That was the time before the prohibition came out from the federal government. But today, we have one person or one laboratory in Todd Vanderah's laboratory that is actually investigating the receptor system for the endocannabinoid system that serves to receive the messages that cannabinoids and other compounds provide to the system. And if I might say so, Todd Vanderah is responsible for all of us sitting here wanting to use cannabinoids because he knows all about where they go and what they do.
All right, I get this one here. Thanks.
Okay.
You can hold on that one. Okay, well, thank you, Rafi. And I want to thank Dr. Gruner for actually organizing this symposium. I think it's well overdue. And I'm excited about having a whole day in which we can actually talk about cannabinoids. I also want to thank all of the deans and institute directors and everybody that's actually supported the symposium. And finally, I want to thank all of you for being here. And hopefully we'll learn a little bit about cannabinoids. And so I'm going to talk a little bit about a few different things this morning. I'm going to talk a bit about endocannabinoids and give maybe an overview, first of all, about the cannabinoid system that we know of. I'm excited about understanding the endocannabinoid system. I think it's the newest part in understanding cannabinoids. But I'm also going to finish with a lot of the work that we've done. We've actually been working on cannabinoids here at the U of A for over 15 years. I started looking at cannabinoids probably when I first came back after doing a postdoc, trying to figure out would they be helpful in things like chronic pain and inflammation. And so I thought I would start with just a little bit of an introduction about cannabinoids and cannabis. And I'm not sure if we have a light that can turn off on the front. But anyway, we'll talk a little bit about cannabinoids. And what I wanted to actually do was to talk a little bit about where the research in cannabinoids are going. And so, of course, the most important part of this is to understand that things like medical marijuana have many different constituents or chemical components in it. And I think we're only beginning to understand all of the different types of chemicals that can be found in cannabis. And so, again, I just point out that people have suggested that there's over 500 different types of constituents within medical marijuana. But for us to really understand some of the science, we have to get it down to the unique compounds. Now, most of us know about some of the compounds that are in medical marijuana or cannabis. Of course, THC has been the one component, or tetrahydrocannabinol, that's been studied a lot. And so if you look in the literature, there's a ton of papers on THC. But after that, it really kind of weans out. There's not a lot of studies on many of the other components. And again, there's over 500 components in there. Over 100 of them are referred to as cannabinoids. But really all the data and research has been focused on THC. And it's THC that we know of is the component that will produce the psychoactive components, or even possibly the euphoric type components. But there are many other components that are in cannabis that we know of that are not psychoactive and don't produce the euphoric effects. But we just don't know a lot about the particular compounds. Now, of course, CBD has been a hot item, so I think we've probably all across the country have seen the commercials. Last year, it was predicted that it made over $1 billion, so obviously there's a lot of people with interest in looking at things like CBD and its therapeutic effects. And I still think that today we know very little about how CBD works. I'd love to come here today and tell you exactly I know how it works, but I don't have that answer quite yet. There are other types of different cannabinoids in the actual compounds, and I think we need to do a lot more study in understanding this. Now, I've brought some chemical structures, and the only reason why I did this is because I don't want to get into chemistry. But what I want to make a point here is that these structures are very similar, but do very different things. And so when you look at something like THC, the one that we've known a lot about for many, many years, it's very similar to the structure of CBD. And so the only thing that's different is this ring right here, where you have a full ring with an epoxide or an oxygen in there, and you look here and it's actually broken. That's the only real difference between the two. However, we know there's a big difference when you look at these two things individually, let's say in animals or humans, in whether they're going to produce psychoactive components, what receptors they work at, and what they might be therapeutic for. And so it's amazing that you can have such a small little change, but you can have a very big differences in therapeutic use. When we look at some of the fronts of where the research is going in cannabinoids, we can think of that people are trying to make more potent and more selective components of cannabinoids, not only just using medical marijuana or understanding what's in there, but actually looking at how we can sort of isolate these compounds and understand their effectiveness. And so when you look at it, we know that there are a number of different both academic institutions and pharma institutions that are actually making what we call peripherally restricted. This simply means that they're going to make compounds that don't get into the brain. And so we know that the psychoactive component is occurring because it gets into the brain, and so many companies have actually made these types of compounds in which they don't cross the blood-brain barrier, but they still have an effect in the periphery. And so I myself, working with a group at UCLA, have actually published a recent paper showing that we can actually use these kind of compounds and we don't get the euphoria, you don't get the psychoactive component. We also have been looking at CB2 agonists, and unfortunately, I think few studies have actually studied this particular compounds that act at this receptor, and I'll talk about the receptors themselves. But I really love this particular area, and I'm going to show you why, because we have compounds that are very effective in things like metastatic breast cancer. And so I'll show you some of that data as I finish up my talk. They're also looking at compounds that are mixed compounds, and then this is really the hot new area. This is endogenous cannabinoids. And again, these are fairly new in the fact that our bodies make cannabinoids. It's the same thing that people knew maybe 50 to 100 years ago about opiates, right? So when you first heard about endorphins and the runner's high, that your body actually makes an opiate. Well, we also know that the human body makes endogenous cannabinoids. And so what's interesting about these compounds is that they're lipids. They're a little bit different than, let's say, some of the peptides like an opiate or a neurotransmitter. These are actually lipids, and they're actually being made out of the membranes of many different cells. Many cells are actually capable of making endogenous cannabinoids. And we're just beginning to learn about these particular structures. I put on here in the fact that companies and academic institutions are very interested in making these inhibitors. What are these inhibitors? Well, they're enzymes, things like MAG lipase or this FA inhibitor. These are enzymes that break down the cannabinoids. So your cells will make the cannabinoid, and then we have enzymes to degrade those. And I'm going to take you through a few slides on that. And the point is that if you block these enzymes, as they're called inhibitors, you can actually increase your own cannabinoid. Okay? And so there's a lot of different academic institutions and pharma companies that are interested in making compounds here, and I'll show you why in a couple slides here. And so when we look at the cannabinoid receptors, what do we know so far? It's identified that there's been 2 cannabinoid receptors. I believe there's more coming, especially understanding how CBD works. But right now we know that there are 2 different types of cannabinoid receptors. And the one that's been studied the most is referred to as the CB1 receptor. This is a G protein coupled receptor. It's an interesting receptor in the fact that some people suggest that it's the highest concentrated receptor in the human brain. I don't think anybody's found anything different in this, in the fact that this is a very high concentrated receptor in the human brain. It's found on neurons. It's known to actually interact and allow for synaptic plasticity or changes within the synapse, but I still think we don't know near enough information about how it's affecting neuronal communication. However, there's a lot of CB1 receptors in the human brain. They're also in the peripheral nervous system, anywhere you look, and when you look at any of the different organs, what people will find is that they seem to show up in almost every organ that people begin to study. So there's a lot of CB1 receptors that we know in the human body. How about approval? Do we have any medications for approval? Well, we do know that the FDA has approved at least 2 different types of compounds that act at the CB1 receptor. So this is nabilon and dronabinol. They are CB1 agonists. They're selective compounds. They are approved for things like pain, chemotherapy-induced nausea, as well as improving appetite in HIV and AIDS patients. But I also put on there that they are known to produce psychotropic effects. We know that, and we know that compounds that act at the CB1 receptor will produce these psychotropic effects. The CB2 receptors are really sort of still unexplored. The CB2 receptors tend to be more on immune cells and glial cells. So the glial cells, things like microglia and astrocytes in the brain, are things that are helper cells, and or the microglia are like the immune cells in the brain. And so it makes sense that they're actually on the glial cells. I put an asterisk on here. Some people report that they may be on neurons. Some people say no, some people say yes. We do know that they don't produce the psychotropic effects that CB1 agonists do. And some people suggest that maybe they're expressed on neurons after pathology or changes. And so there's lots of investigation to determine whether they do show up on neurons. But we do know they're on a number of immune cells, spleen, bone, colon, liver, pancreas, and they tend to be on the immune cells of those organs. So many organs will have sort of their resident immune cells, and that's where you'll see the CB2 receptors. Potential therapeutic use, of course, is pain and inflammation, an area where I've worked on for many years for CB2 receptors, but also osteoporosis, and I'm going to share a little bit of work on that, knowing how CB2 agonists may affect bone and bone remodeling. And the important part is the non or no psychotropic effects. And the only reason why I point that out is because, as I said, I started working on cannabinoids about 15 to 20 years ago. And in order to get money from the National Institutes of Health, I had to work on something that was a little bit less psychotropic than the CB1. And so I had sort of a ploy to say if I looked at something that was not going to produce psychotropic effects or possibly be rewarding or lead to addiction, could I study something that might have an effect on pain? And so my work, again, started a long time ago in this particular area of CB2 receptors because it lacked psychotropic effects. So a few things I wanted to ask. So can we increase endogenous cannabinoids? And what are they used for? What could they possibly be used for? And I'm going to show you a few things here. But I also like to always correlate this with this comment. And I think it's important today that we continually have this discussion to understand that many, many things will not only produce a medicinal therapy, but they will also produce unwanted side effects. I think as a pharmacologist, I know that every single medication out there will produce something that we like, but it can also produce things that we don't like or unwanted side effects. And I think it's important that as we go forward as a community in studying cannabinoids, that we do both of this to be responsible, to understand what are the good and what are the bad. And so I've pulled a few papers. I love to kind of look through the literature of what's happening in the cannabinoid field. I love to pull really recent papers. This was a recent paper in 2018. It's a beautiful review that has tons of different studies, both clinical and preclinical, so human studies and animal studies. And it wanted to ask the question of where do these endocannabinoids coming from? And when you look through this particular review, you'll notice that it comes from many, many different cells. Any cells that have the enzymes to make it will have the ability to produce endocannabinoids. And so when they had a picture in this particular review, and it talks about the idea of where are they coming from and what's inducing them. And you'll notice when you look on this side, the green is sort of showing where are they increased and what stimuli. It's often stress. Stress, pain, certain types of food, circadian rhythm. So there's an up and down of endogenous cannabinoids based on circadian rhythms. So, and you'll see that a lot of these things are producing the endocannabinoids. It goes into your circulation. And then, of course, they propose that it's working at these different receptors, whether it's CB1 or CB2, and it may have some type of effect. What was interesting that was pointed out in this particular review over and over again is that endocannabinoid signaling can increase consumption and storage of energy. So this was even new to me, but I thought this was quite interesting in the fact that it can do this, and that just like endorphins, that physical exercise can actually mobilize your endocannabinoids. So actually exercising was shown in a number of studies to actually increase the amount of endocannabinoids in circulation. It's also important that the review pointed out that some endocannabinoid signaling may restore homeostasis following stress, and that stress can release it, and that actually having increased levels may actually decrease some of that stress. So let's look closer at the endocannabinoid system so that we can all kind of understand what's happening here. And so I've just put together a picture here. If we're looking at some cell within the body, and again, endocannabinoids are made in many different cells, and we're still finding out what cells are producing them. If we look at the outside and the inside of a cell, I've put my cannabinoid receptors here, and again, these will act at both CB1 and CB2 receptors. And again, these are G-protein coupled receptors. When we look at the endocannabinoid system, we realize that it's actually just being made from the lipids of the membrane, just like our prostaglandins are. And so when you look at it, you see that there's very common enzymes. Phospholipase C, boy, this is a very, very common enzyme in many, many cells. And then DAG. So these are the two enzymes that produces one of our most abundant endogenous cannabinoids. and that is what we call 2-AG or 2-arachidonylglycerol. So this is the one that's probably the most abundant. I've actually put reference, or I made several different papers suggesting the fact that it's 200 times more abundant than the one that was first discovered, which was anandamide. But these are both lipids, very similar in structure, both endogenous cannabinoids. And again, we think of 2-AG now as being the more abundant endogenous cannabinoid. And what's interesting is that when they're made, they can act at the CB receptors. Some people suggest maybe from the internal side, some from within the membrane, and some think that these cannabinoids, which we do know, can get released and actually act back on the receptor. And so these cannabinoids can be made, released, and act at CB2 receptors. Again, the same type of scenario here, in which, again, people are studying the enzymes for the production of anandamide. What's even most interesting in this field, and where a lot of people have started to make compounds for this, is at these enzymes. So I put these in this sort of this pink or reddish color to suggest these are what's actually breaking down the cannabinoids. So these are the enzymes that sort of remove it from its activity. And so far for 2-AG, there's really been 2 different enzymes that have been discovered so far, and they will break it down. And I think what's very interesting when you look at the breakdown product, it's arachidonic acid. Arachidonic acid, then, we know, is acted on by the cyclooxygenase enzymes to produce prostaglandins, which again are things that we think of as pain and inflammation. And so you see a similar type of pathway for the anandamide. And so a number of companies as well as academic institutions have started to look at the idea of blocking these enzymes. Okay, so there are a number of new compounds that are going to come out. I'm going to show you some data that I've used with one of these that actually will block these enzymes. And you can see that there's 2 things that are going to happen if you block these enzymes. One, you're going to increase your endogenous cannabinoid. And number 2, you're going to decrease the production of prostaglandins. And so in that case, then you would have sort of 2 different ways of having something that might help with pain and inflammation. But you'll see there are a number of different studies that are looking at these enzyme antagonists that will be used for things like epilepsy, traumatic brain injury, Fragile X, and encephalitis, as well as some different types of multiple sclerosis. So all of these studies are sort of ongoing with these new compounds that are coming along that I think we should all be aware of within the next year or so. The other thing that I find fascinating about cannabinoids, because again, we haven't known about this system for very long, is the idea that it's very highly influenced by a very, very common type of receptor, another G protein. They couple to what they call GQ, and these guys directly will activate this pathway, the phospholipase C pathway. So it's interesting that we've had, and I just listed all of the receptors, and these are not even all of them, I should say, but many of the receptors that are all coupled through what we call a GQ pathway. All of them. So even things like Benadryl, that's blocking the histamine receptor that we use to actually go to sleep. But the point is that they're all influencing the levels of endogenous cannabinoids. And until we knew this pathway, we didn't know that all of the medications that are acting at all these receptors are also influencing this particular pathway, whether it's increasing or whether it's decreasing it. And so, again, I think that there's a lot to be studied, both with the cannabinoid system, but also how other medications may be affecting this particular pathway. The next is to understand that these enzymes are regulated by a number of different things. So first of all, we know that there's different expression levels in different tissues. So some tissues make a lot of these enzymes, some make very little. We also know that they'll change expression based on sex differences, so whether estrogen or testosterone's there. We know that there's big differences in swings between the production of endogenous cannabinoids. And finally, there's many studies now that are looking at pathology. So what happens in a pathological state, in a state of cancers or lupus? And so they're looking to say, are there changes in these enzymes, and therefore changing the endogenous cannabinoid system? And so I find it great interest in understanding how these enzymes are influencing the levels of endogenous cannabinoids. The other really interesting data that are just coming out now is to understand that they may influence channels. So I, of course, have the cannabinoid receptors that we know they're working at, but there's great interest in understanding whether endogenous cannabinoids, and I'll throw in here maybe CBD, is maybe acting at a chloride channel. Now this is important because we do know that CBD, there's a compound called Epidiolex. It's a CBD agonist. It's been approved by the FDA for seizures. We know that many of the seizure medications will affect chloride channels to sort of decrease the excitability of neurons. And now there's great interest in a few different studies that have shown that 2-AG, our endogenous cannabinoids, may actually allosterically modulate this chloride channel. And so again, I find great interest in seeing more research done in this particular area. Okay, so I brought along more clinical type papers instead of preclinical, and I wanted to sort of say, have people really started looking at endogenous cannabinoids in clinical studies? And this was a wonderful study, again, a very recent study, 2019, in which it looked at endocannabinoids in obsessive compulsive disorder, OCD. And I won't take you through all the data, but I love this actual picture, because in OCD, it's one of these conditions in which individuals will have a lot of thoughts. We know that thoughts are driven out of the frontal cortex in many cases, and so you have a lot of thoughts, and it drives it down through what we call the basal ganglia. This is a part of the brain that allows up for a circuit to start to happen. And so people with OCD will have a lot of thoughts, and the thoughts will keep going around and around. And what we think of is it sort of goes as a thought into the basal ganglia and then back to the cortex. Now, the thoughts, most often in people with OCD, will be something of maybe I have germs or bacteria of germs. And so what they do then is then they have a motor response. The motor response will be, go wash my hands. I got to wash my hands over and over again. And that's what we think of as a motor reflex that, again, goes constantly through the basal ganglia. That thought of the bacteria, then the repetitive of washing, then somehow causes a relief. Like, okay, now I'm away from the bugs. And that we think of as sort of the limbic system, sort of this primal system that says, okay, I've done something to take care of the possibility that I may have bacteria on my hands. And so it's a repetitive process that often people with OCD will go through. And what's interesting in this particular study is they really looked at both cannabinoid receptors and endogenous cannabinoids in this system. And like I said, of course, you'll always find the CB1 receptors. They're everywhere in the brain. There's a lot of them there. But they're heavily focused in areas of the basal ganglia, whether you look at the putamen or the globus pallidus. So there's lots of different areas, but they're heavy in there. You'll also see they're very heavy in the cerebellum. But they're in there, and they also noticed that the endogenous cannabinoids would be there. And so in this particular conclusion of the paper is, and again, this is both a study that was looking at humans and animals, in which they looked at neuroimaging, and they found that, again, the endocannabinoid system would sort of reduce the amount of anxiety and compulsive repetitive behaviors in these particular studies, both in the model of animals of OCD as well as in humans. Again, the greatest thing about these types of studies is they suggest there needs to be a lot more work done here. Again, I think they were just claiming that they saw something, but I think the important thing is that there are call for more studies of looking at the endogenous cannabinoid system in things like OCD. Another wonderful paper, this is looking at whether endogenous cannabinoids play a role as neuroprotective due to oxidative stress that we know produces this neurodegeneration diseases. So we do know that the overproduction of reactive oxygen species and nitrogen species will result in some cases neurodegenerative diseases, so not always. But we know that when you increase these things, you can have an increase in the degeneration of nerves. Again, not always, but we do know that. And this is a group actually that comes out of NIH in which they've shown that there may be an endocannabinoid system that may actually play a role in decreasing some of these reactive oxygen species. And so I've put a picture up from this actual article in which I love this picture because it sort of shows the fact that CB2 receptors may be responsible for some of this decrease in inflammation and decrease in reactive oxygen species. And so you'll see that, again, the CB2 receptors tend to be more on these cells. Again, these are more the glial cells and the immune cells of the CNS. They may be also on they are on inflammatory cells that may get access into the CNS. There's question marks of whether CB2 is on neurons, so it's nice that there's question marks, but there's also some other cells where you'll see the CB2 receptors. And again, it's thought that when CB2 receptors are activated, it will actually decrease things like oxygen species, nitrogen species, cytokines, chemokines, and so it tends to decrease that inflammatory factors. And so with this in particular study, or this review, they were suggesting that maybe endocannabinoids may help with neurodegenerative disorders like stroke, traumatic brain injury, Alzheimer's, Parkinson's, and MS is what they actually listed in the paper, suggesting that it may be helpful. But there are other studies. This is a very recent study. This was just published in JAMA in Psychiatry in 2019, in which they also suggest that cannabinoids can be dangerous. And so I love the idea that we have to think about the fact of what's happening here on both levels. In this particular study, again, this was a study done in humans. They looked at actual imaging, and they looked at certain channels like TSPO. This is a channel that's found on immune cells as well as macrophages and also on mitochondria. They've also looked at other types of inflammatory factors, so I just put inflammatory factors here. And they showed that cannabis users had an increase in these inflammatory factors, suggesting, again, in this particular paper, that it may be through more of a CB1 receptor, which we know is heavily concentrated in the brain. We know that it's on the neurons, and that may be doing sort of the opposite of the CB2. Again, and suggesting the fact that there was this increase was also due to some chronic stress and anxiety, which the people that were using the cannabis were also having. So again, I think that we need to have more studies to sort of determine what's the good and bad of CB1 and CB2, and whether there's truly inflammatory type factors that are being developed here. Another wonderful study here, again, it was a study that was done in humans. This is an fMRI study in which they're looking at the areas of the brain that are active during the use of either THC and or CBD. I think as time goes on, we'll start to see more and more studies where they're combining the different types of cannabinoids to see what their outcome would be. And so this was a wonderful study in which the conclusion was to simply show that THC, which again is more of a CB1 agonist, would disrupt the default mode network. Now I find this interesting because this is the network that's sort of our underlying basal activity of the brain. Some people will say it's your daytime dreaming mode, or it's the mode that sort of sits there as your baseline level before you're really going to do things. I believe it plays a big role in a number of different decisions that we actually make. It involves areas like the cingulate gyrus, which is part of the limbic system, so I do believe it's sort of a decision-making area. But what they found was that THC really disrupted that mode. So they did this in individuals that weren't using THC. They got their modes and all their imaging, and then they put them on THC, and then they actually did the imaging again, and they saw that there was a disruption of that. I don't know what that really means, except for that there was less function, possibly in that mode. CBD didn't do this when they did CBD, and what was most interesting in this study that I'm seeing a lot of papers now is that CBD sort of restores the default mode. So it's interesting, when they combined the two and they gave them CBD, it actually restored the disruption that was being produced by THC. So the combination of the two sort of took things back to baseline. And I find that very interesting, and you'll see more and more of these types of studies. And so again, I always like to talk about the fact that cannabinoids, we can't just say that they're all great, and we're going to talk them up here and tell you how wonderful they are, but I think there's lots of things that we need to do to make sure that we're looking at whether they're good and bad together. So again, there's a study that came out here in 2019 suggesting that they may have long-term impact on the brain and that they can produce some psychotic symptoms and cognitive effects. And I think most people would tell you that they can do this. It doesn't mean it's going to happen in everybody, but they can. Again, this particular study found, and I love the idea that now they're doing studies where they're combining the drugs, but when they combined it with a CBD, they actually restored it, and they attenuated the psychoactive components and the cognitive effects. This was in humans. This is also confirmed in animals. And so again, I think we're going to start seeing more and more of these types of studies where there's a combination of the cannabinoids. And so there's a ton of different papers out there. I can't go through them all, but I thought what I would bring this morning is a little bit about what's happening in the cannabinoid field as far as clinical trials. And so if you go on to the National Institutes of Health and you can look up clinicaltrials.gov, you can see what's active today as far as clinical trials. So these are actually ongoing trials. And you can see that almost 200 clinical trials are ongoing with CBD. So this is the big one, really. And I've just listed some of the indications they're going after. This list is quite long, and actually it duplicates. There's a lot of studies with epilepsy. But look what number 2 was. Drug dependence. I find this just fascinating. And the fact that you're using a cannabinoid to help other people, and these are studies that they're looking at for people who have drug dependence on alcohol, on benzos, on amphetamines, and even opiates. And so it's interesting that they're going to use CBD in a clinical trial to determine whether that will help people with drug dependence in other areas. And so we've come a long way from claiming that cannabinoids are the gateway of drugs of abuse to the point where one of the cannabinoids might be used to help people with drugs of abuse. And again, you'll see the full list there, and I can't wait for the next couple years when these trials finish to sort of find out what was effective, what worked, and what didn't. If you look at medical marijuana, there's currently 44 clinical trials ongoing, and again, the list is very similar. And so for CB1 agonists, these are ones that are selective now. For CB1, you have about 14 ongoing current clinical trials, and in CB2, only 10. So again, I think this is the area where I'm going to show you some data that I think is understudied, and we really need to start investigating. Okay, so many of us know, and I work in the area of pain. I've worked at chronic pain for many years, trying to figure out how we could develop new compounds to help people with pain that weren't opiates, that weren't addictive. And so we've done this work, again, for over 20 years now, trying to look at things that might be helpful. And so one of the areas we got into was cannabinoids, of course. And I'm just going to show you some clinical things in which, again, it's been shown that smoked marijuana is effective in things like chronic neuropathic pain. So you'll see a lot of these studies. This is just one I chose. There's also vaporizing of marijuana can be effective, and we know that they do help people, not everybody, but some people with neuropathic pain do get relief using these types of either smoked or vaporized marijuana. When you get down to the individual compound like THC, the data definitely vary. So there's a take-home message here is that when you have something that's sort of a mixture, remember, we're looking at things here with over 500 different chemical constituents, are more effective. When you get down to the individuals of THC, and years ago we actually ran a study here with THC or dronabinol, and you get mixed results in all reality. And I do believe that with THC, you sort of have a very strange dose-response curve we often talk about, where if you increase the dose too high, you actually kind of table off and it starts coming back down, and it actually is not very effective. And so there is a lower dose that tends to be more effective, but you start to reach that point where you'll start to see many of the patients that will start complaining about cognitive impairment. I don't feel good. I don't like this. We had that when we did our study here. Many of the patients were like, yeah, maybe the pain's gone, but I don't like the way I'm feeling right now. And that was very obvious when we did it with, again, a pure THC. So there's something about the idea that maybe the mixed, many chemicals within the marijuana might be a little bit more better, or might be better for people with neuropathic pain. Again, you'll start to see many more studies now that come out with mixtures. So whether you have a 1-to-1 ratio of THC to CBD, again, you're starting to see that there is efficacy in clinical trials, reducing pain and spasticity with no tolerance, a chemotherapeutic-induced neuropathy model in which, again, a small number of patients, but you'll start to see positive results when you have mixtures of the compounds. I also want to always look at the psychiatric component of this. And again, there are a number of different studies. I could have picked a bunch, but when you start to mix the two, you can sort of start to reduce the amount of psychoactive components, just as I mentioned in that earlier study. So when you start to mix things like THC and the CBD, we do see that there's a decrease in the amount of psychoactive component that's actually being generated. And then, of course, there are a number of different mixtures of cannabinoids, whether it's CBD as well with CB2 and CB1 agonists, that are very effective for things like Crohn's disease or IBS. These are very inflammatory and painful type disorders. And so I thought I would bring a study. I love these type of studies that are randomized, placebo-controlled, double-blind. This is a beautiful study in which, again, they showed that these cannabinoids were effective for these patients who are suffering from things like Crohn's disease. And finally, I went to, and I've talked with Arena. This is actually public data now. They've come out with a compound that tends to be more selective for CB2 receptors. And again, I've been encouraging many of the pharmaceutical companies to kind of go down this alley. Again, there's not one that's approved yet by the FDA in patients, but this is a leading compound. It's a CB2 agonist. And they did it in a small clinical trial. This is a positive phase 2A trial, again done by Arena, in which they showed that it was effective in Crohn's disease. They only had 14 patients here, but the outcome was really good. And they basically stated the fact that there was significant decrease in improvement of pain. I looked at the pathology, and the pathology was amazing. And so I really do believe that the CB2, we hopefully will see they're now going to be running a larger phase 2 clinical trial with their compounds to go forward. But I do believe that this is a great area for something like cannabinoids. And so I'm going to transition to a little bit of the work that I've done, and hopefully we save some time for some questions. So I've looked at CB2 receptors, as I said, for over 15 years now. And again, the reason why starts here at the bottom, really, and that is that CB2 receptors do not produce the psychotropic effects. So I was lucky enough to get a grant from the National Institutes of Health in the area of cancer, of all things, because again, a lot of the cancer patients will suffer from chronic pain, and the only thing that we have for metastatic cancer patients for pain is opiates, and those doses get ramped up really quick. And so I was able to convince them to fund some research that we had. We've known prior, many of our studies have actually shown that CB2 receptors can relieve inflammatory and neuropathic pain, and so we used that as evidence. And then there were CB2 receptors that are not only on glial cells, but also on osteoclasts. This is a wonderful group that was over in Israel, in which OFEC and BAB, their group, actually showed that maybe CB2 agonists would be good for osteoporosis. And I thought, well, perfect. Why don't I look at these things in metastatic cancer pain? And the reason why is because within the bone, and if you're looking at bone here, you have really sort of three major cells. You have the osteocyte that sits inside the bone, and it can become an osteoblast. Osteoblasts build bone. And then you have these cells. They're much bigger. It's like an immune cell. It's like modified macrophage, and it's an osteoclast. And this is what actually destroys the bone. And we know that cancers, many of the metastatic cancers like breast and prostate and lung, they'll go to the bone, and they'll activate these cells. And these cells will start to degrade the bone. The reason why we think it's happening is because there's lots of growth factor that's also stored in the bone, and the cancer is smart enough to figure out how to get to that growth factor. And so we know that cancer will activate these, and we know that in many of the different diseases, metastatic cancers, that you do have bone loss. But these cells also contain the CB2 receptors. And so I wanted to really figure out whether it would be effective both for the pain and what would it do for the bone. And so we had to come up with a model, and so we used this model that was first developed at the University of Minnesota, in which it was looking at a metastatic model in which the animal doesn't get too sick, but most importantly, it's a model that allows me to detect pain. Remember, when I'm trying to find out if an animal's in pain, it's very difficult because I can't just ask the animal. I can't say, here, give me a mark between 1 and 10. And so I have to figure out a way. And so what we do in the pain field is we look at ways that they respond to stimuli. And in this particular case, if I anesthetize a mouse and we move over the kneecap here, and we find the distal end of the femur, we drill a small little hole, and I can inject the cancer cells directly into the femur. And then I can plug that femur, and for the most part, the cancer cells will pretty much stay in there and continue to grow until they eventually fracture the bone and then start to leak out. And so in this particular case, I used breast cancer. So we used a breast cancer model. We put them into the femur. We plugged the femur. And within about 7 to 10 days, you will see that the animal will start giving me signs of pain by either holding that paw up or continuing flinching that paw. And I can use that as my marker that the animal's in pain. And so these are just some of the data that were collected by Alicia, who was a graduate student at the time. She's now working at Vanderah. And so here you can see this is the amount of pain. So this is the number of flinches. And as you go up, you get more pain. At baseline, of course, none of the animals have pain. We then do a surgery in which some of the animals get the breast cancer cells and some get the surgery with the media, but no breast cancer cells as our control. And then they all will either get a vehicle or a different type of cannabinoid. In this case, I'm using CB2 cannabinoids. And you'll see by day 7, as the cancer continues to proliferate and grow in that bone, you'll start to see that the animals express pain. And so that's shown here by these 3 groups. And so this would be, at this point, maybe a metastatic cancer patient who comes to the doctor and says, I'm having pain in my back or my hips. And so at that point, then we started to actually dose with the cannabinoids. In this particular case, they're going to get either a vehicle or 2 different types of cannabinoid. And we just gave them a dose once a day, 6 milligrams per kilogram. But you can see by day 14 that we actually had a significant reduction in their pain. So this is the vehicle, and you can see that the pain is increasing. If we go out to day 21, it just goes up. But the cannabinoid actually significantly dropped that pain. So it was decreasing the pain in these particular animals. And we did a bunch more studies, but I'm just going to show you what happened to the bone. This is always very interesting because I wanted to know, were we affecting the actual bone? And so if you start over on this side, you can see the cortical bone here. This is a normal femur out of a mouse. Again, you get nice cortical bone, you get trabecular bone, nice thickness when you look at the cortical here. This is the model. This is our cancer model, so this is day 14. And again, you can see that there's a lack of cortical bone thinning here. You see a nice fracture right here. You see a loss of the trabecular bone, and you even see a little bit of a growth on the outside. We know that breast cancer can be osteolytic, which breaks it down, but it also has an osteoblastic component where it builds a little bit on the outside, but it's non-structural. This is what it looked like with the cannabinoid treatment. Again, we started on day 7, so they've only received from day 7 to day 14 cannabinoids. And it wasn't perfect, but it does look better as far as its trabecular bone, as far as its cortical bone, no fractures and less growth on the outside. And so these were some of the first data that we actually determined that maybe CB2 agonists are being useful in inhibiting bone loss in this particular model. And so we can do all kinds of measurements because in order to get it published, all the journals want you to do a gazillion measurements. I'm going to show you just a few, but we actually look at bone volume and trabecular volume, and you can see in the model that goes down. You can look at bone mineral density, and again, in the model it goes down. But when you put on the cannabinoids, as shown here, you continually sort of decrease that bone loss. We looked at the activity of the osteoclast. That's all this measure is. This is something that we actually do in our patients here at the U of A. We monitor their bone loss by just pulling serum, and you get measurements of osteoclast activity and also the collagen that comes out of the bone. And so we did the same thing in our mouse model then. And again, you see that there's an increase in osteoclasts. These are the ones breaking down the bone. And you also see an increase in that collagen that's coming out of the bone as it gets destroyed. And in both cases, the cannabinoids were decreasing that bone loss. We then finally looked at fracture. And of course, the ultimate thing is when do they actually fracture? And so this is just the percentage of mice that fracture. These data are all done by x-rays, and they're given to a radiologist in which they are blinded, and they just score how many fractures occur within each one of the animals. And you can see that the animals that didn't get any kind of cannabinoid, just vehicle, over 50% of them at day 14 were already fractured. Whereas in the cannabinoid treatment, again, both of them are reduced. It wasn't perfect, but we were down about 30% with this cannabinoid and even lower, down below 20% with this particular cannabinoid. And so they were actually decreasing the number of fractures. One other thing we had to measure is whether there was growth of the tumor. And so we looked at the tumor growth. So this is a pathological stain, an H&E stain. And again, you can see that this is pretty amazing, but we had no idea what would happen with the tumor. But we actually saw that the tumor was slowing in its proliferation. And there's multiple reasons why, because as I said, what's in the bone is growth factor. If you're decreasing the amount of growth factor releasing, you're probably also going to slow the growth of the tumor. And that's what we saw. So again, the lighter colored cells are actually the tumor cells. So these are the breast cancer cells. The darker ones are hemopoietic cells. Those are normal. And so you can see that in vehicle, it was about 72% occupancy of the actual tumor cells, whereas the ones treated with the cannabinoid were significantly reduced based off, again, pathological analysis. And so that theory then was picked up by another student in the lab, Kat, who said, if this is the case, and if it's true that cannabinoids might slow proliferation of the tumor, let me try taking these breast cancer tumors and put it into the breast of the mouse and measure how big it gets. That makes sense. So she took the breast cancer cells, she put in those luciferase reporters so you could actually see the tumor. When you anesthetize the mouse here and you give them luciferin, and you can see the tumor. So there's the tumor, and you can see that it continues to grow and expand until we have to sacrifice the animal, but the tumor will grow as a primary tumor and well as metastasize. And what she found was that when she gave the cannabinoid, there definitely was a significant decrease in the growth. It still continued to grow, it just was slowing that tumor growth. And so again, this is just one example, but she had many animals. This is a representative of all the animals in which she measured the primary tumor, and she continually saw that it did slow the growth, but again, not like a chemotherapeutic where it destroyed it. We then went on to look at the endogenous cannabinoid system. Again, this is a newer part of the story in which we just started getting new compounds for trying to increase endogenous cannabinoids. So Austin in the lab has been working on this. This is a compound that blocks that enzyme, that MAG lipase enzyme, so it works right there. It's a compound that's been given to us from Scripps, from Ben Cravat's lab. And what it does is when it blocks this enzyme, again, you get that increase in 2-AG. And also remember, it's going to decrease the arachidonic acid pathway, which hence leads to prostaglandins. And so you're getting a two-factor. You're getting an increase in cannabinoids, and you're going to decrease prostaglandins. And so he's tested this in, again, our cancer model as a possible therapy. So here, again, you're looking at flinches and guarding. You're looking at the fact that it will increase the pain, the model itself. So this is just vehicle here in the green. And then you're seeing that when you block that enzyme, you actually get a nice decrease. And then he just gave one dose here, and you see it goes down, and it comes back at about 180 minutes or so. And so the idea that you could actually increase endogenous cannabinoids may also help in metastatic cancer pains. And again, he's shown that after chronic use now, if you do it over many days, again, you still see a very nice decrease in that pain. All right, so if we look at sort of a model of what's happening, we believe that, again, in situations of cancer, we get all this stuff happening. We get the idea that there's all kinds of inflammatory factors, cytokines, chemokines, increases in osteoclasts that chew away the bone. Of course, you get all kinds of pain and inflammation that are occurring in metastatic cancers. When we give the cannabinoids, we tend to sort of reverse this. And we can either give cannabinoids, the CB2 cannabinoids, or something that will increase endogenous cannabinoids. And so this sort of flips over, in which you start to decrease all of these factors, and you tend to decrease the number of osteoclasts, the bone loss, the inflammation, and the pain. And so that's how sort of our working theory with how cannabinoids may be affecting metastatic cancer pain. The last part of the talk, I just wanted to share with you a little bit about the idea that we've also looked at it in pain. And so this was a timely conference to talk about sort of cannabinoids, morphine, and pain. This was a poll that just was done by NPR here, August 21st, 2019. You can see that nearly 1 in 5 Americans say they often chronic pain is going to interfere with their lives. And this was 3,000 people that were polled. And so pain is a serious condition. It's a serious problem. And so we started to look at whether we could combine the two. We figured, you know what, opiates are not going to go away tomorrow. And the idea in the lab was that could we combine or at least reduce the doses of opiates by adding on a cannabinoid? And so we did. We looked at the combination of CB2 compounds with opiates in models of chronic pain and inflammation to see if we could reduce that effect. And so again, another student in the lab, Shanice, said, you know, I'll go ahead and take this task. And so she did. She did many different models of injury, chronic pain injury. And so here you can start to see this is a chronic pain inflammatory model in which she's looked at morphine, getting a nice dose response curve. She's looked at the cannabinoid. And then when you combine the two, it shifts to the left. That was a great sign because that suggested that you could combine the two, lower the doses, and you might get a much better analgesic or pain relief effect. So she does a isobologram, and of course this would be the line of additivity when you add the two compounds, and it was always synergistic. So I won't take you through that, but the point is that if you put the two together, it's not just additive. You get synergy, and you get a nice reduction in that pain. And so she's done that in models of neuropathic pain, and again, you'll always see the curve shift to the left here, whether you're touching thermal, you're making a thermal or a mechanical type of assay. She then went on to say, well, if you can put the two together and you can reduce the amount of pain, if I put the two together, will I get more side effects or addiction, or can I reduce the amount of addiction that opiates produce? And so she did a test that's called condition place preference. This is a nice, easy test to determine whether compounds are going to be addictive or not. It's an assay that is basically a chamber in which animals can walk back and forth, but they're different. So this may have a rough floor and a different pattern, a smooth floor and another different pattern, but the animal can figure out which room they're in. And so they know, and typically if you put something like a rat in there, that'll go back and forth, and about 50% of the time here and 50% of the time there. Now, if you simply close off the door and you only allow the animal to stay in one room and you give them your drug of choice in that same room for 2 weeks, right? So if I take it and I close off the door and I put the animal in here and I give them morphine for 2 weeks, and then on test day I open the door, don't give them any drug, typically if it's addictive, they're going to stay in that room more often. And it's interesting, you'll watch the animal, they'll stay in that room. Once in a while they'll run over, they'll see what's in here, and they'll come back. And so typically all drugs that are addictive will do this, what we call a positive condition place preference. And so you can see here, this is for morphine, and of course morphine is producing this positive effect. It's significant, suggesting that the fact that the animal keeps going back to that same room. The cannabinoid does not do this, the CB2 cannabinoid does not do this, but what she found was that when you combine the two, again, the CB2 agonists tend to decrease the opiate effect. And so the animal no longer would go back to that side when you combine the two, and it would go back and forth. And so in order to, again, get this published, we were required to actually measure dopamine. And of course, dopamine is the neurotransmitter of reward. We know for a fact that every drug that is abused will produce an increase in dopamine. And so you can see if we measure dopamine in the brain, you can see that in blue here, again, morphine causes a nice increase in dopamine. And again, the dopamine is thought to be the rewarding effect. The JWH in green does not do this, the CB2 compound, but when you combine the two, we actually decreased the dopamine release. So this was really confirming that a CB2 agonist may actually decrease the rewarding effects of an opiate. And that reminds me to go back to that idea. Remember when we looked at the clinical trials that are ongoing, CBD is being used also as a drug that might help with drug dependence. And so CB2 is another area in which it may be acting to reduce drug dependence. Now, this was also shown by a group at NIH and was published in Nature Neuroscience. Elliot Gardner showed this with cocaine. So he had a beautiful paper in which he showed that certain cannabinoids, CB2 agonists, could actually block cocaine addiction and cocaine-induced dopamine release. And so, again, this was done back in 2011, and then we actually showed it with the opiates. And so I'm going to finish with just a few different studies that we do have ongoing, and then I'll take some questions. What do we have going on? So we have students that are looking at the mechanism of how CB2 may attenuate opioid dopamine release in addiction. So Hong's working hard right now to figure out the mechanism of how that's working. We also have a number of studies that are ongoing to look at cannabinoids in cognitive outcomes. And so we have things like chronic THC, chronic CBD, the chronic CB2 agonist you just heard about, all being given for about 30 days in adolescence. And then we're looking at a whole host of cognitive outcomes. Again, not only looking at the behaviors, but my partner, Dr. Hammer, is also looking at the outcome of what RNA-seq, using that of what's happening in the brain. We're going to look at structural changes within the brain. And so again, my collaborators, Michael Hammer and Tally Largent-Milnes, are actually helping me with this particular study, and Alicia is one of the students, of many students that are actually working on this. The next part of it is to actually take these chronic administration of these cannabinoids and give it to pregnant mice. And again, the whole time during pregnancy, they will be on these cannabinoids. Again, each one of them separate. And then again, we'll do the exact same measurements on cognitive outcomes. There's a little bit of data so far. So it looks as though when we do a chronic CB2, the one I've just been talking to you about for 30 days or about 28 days, we didn't see any change in cognitive behaviors with this compound in the adolescents, so in this particular study. Again, we broke it out between male and female, but we did not see any significant difference in cognitive behaviors with the CB2 agonist. We also have a few other studies going. So again, working with Dr. Hammer, looking at CBD in epilepsy and determining mechanism there. We also have a clinical trial ongoing right now with dronabinol in metastatic cancer patients to sort of back up my work with cancer. I would love to have something more than just dronabinol. I'd love to have a CB2 agonist, but I don't have one yet to go into humans. But we are doing it with dronabinol right now, which acts at both CB1 and CB2. And we're also looking at some natural compounds, the CB2 agonist, this BCP compound for clinical trials for ulcerative colitis and Crohn's. And so with that, I just want to thank the whole group that's actually made this possible. Like I said, we've been working on this for many, many years here at the U of A. A whole list of people, many of them have actually gone on and taken jobs elsewhere. And so we still have a big group that's actually continuing to work on this. I have a number of different collaborators. So again, a number of people that I can count on to help me run the lab. And I also want to thank all the funding that's made this possible. And finally, I want to thank you.
Thank you very much, Todd. I think you can see the complexity of the issues involved here. The last slide, in my opinion, summarizes it best. You have like 30 or 40 people all focusing on this kind of an issue. We're going to have just a few questions, please, if you want to come to the microphones, and go ahead.
Excellent talk. Chris Bannock, assistant professor in physiology. So I study peripheral nerves in cardiovascular disease. And our recent findings suggest that peripheral afferent nerve activity in the kidney is elevated, as well as sensitivity is elevated. Now, when you talk about decreases in peripheral pain, I'm wondering if you've sussed out whether it's tonic activity that's reduced with your CB2 agonist. And I'm assuming that this is just peripheral because it is CB2. Or if it is the hypersensitivity is attenuated.
So you're asking whether the compound, in our cases now for pain, would actually produce what we would call analgesia. Analgesia is something where all of us would sort of take something that, let's say we're not in pain, and we would take it and we would have a decrease in our pain threshold. And that's what we sort of call decreasing even the baseline. And in the case of cannabinoids, when you look at CB1, absolutely. So with CB1, we know they're on the nerves. They act similar to opiates. They're not as efficacious as opiates on those nerves, but they will produce that, so they will actually drop that. For CB2, you tend to have that pain and inflammation. And that mainly makes sense in the fact that CB2 receptors are going to be located on your immune cells, and part of that inflammatory. And so it looks a little bit more like an NSAID. However, it seems to have more of an effect than an NSAID, the non-steroidal anti-inflammatory, where it will actually drop that hypersensitivity effect. but it doesn't really have a lot of the analgesic in there changing that nerve conduction activity.
So in your opinion, the CB2 agonist is strictly, the effects are strictly because of the anti-inflammatory effects? Not
Yes, at least that's what we think. You know, again, we need a lot more research than just mine, but I do believe that it's affecting more of the inflammatory components of that. Now, eventually, when you start to actually change the nerve structure, you may have eventually effects there, because again, all those inflammatory factors affect the nerves. And so they start to phosphorylate channels, and so you start to have a sort of a change of the neuron, and that sort of leads to what we call chronic pain. And so you can actually start to change the actual phenotype of nerves if pain continues on. And so do cannabinoids change that? I haven't done a lot of investigation long term to see what those changes are on channel activity.
I see no further questions, and
just one.
oh,
Hey, Todd.
right.
Great talk. I was curious about the osteoclast situation and how you mentioned it degrades bone. It's a modified macrophage, and what's the connection between calming the bone degradation aspect of CB2 on the osteoclast and inflammation, let's say, with macrophages? Then you have microglia, which are also like modified
Right.
macrophages in the brain.
Right.
Do you see a connection in terms of the mechanism, or is this
Well,
speculate?
no, I definitely think there's a very common mechanism. So it seems like when you give CB2 agonists, and I didn't show all the data, but you definitely get a certain decrease in a whole profile of certain cytokines and chemokines. And again, the question is whether those are coming from osteoclasts or macrophages. A lot of those actually come out of your immune cells. And what's interesting, when it comes from a macrophage or immune cells, those are what actually then go out and then stimulate the osteoclast. It's very interesting that the osteoclast will respond to those cytokines and chemokines. It activates them, and that's what then causes more bone degradation. And so there's definitely a correlation between the two. Microglia, I don't know. I mean, obviously there's many studies that you'll look at that you'll show that cannabinoids are definitely affecting the microglia, the astrocytes, especially for CB2 in the brain. CB1 as well, but CB1, then you have the difficulty that a lot of it's on neurons too. So,
Yeah. Thanks.
thank you.
Thank you, Dr. Vanderah. I guess my question is generally with the history of the origin of the CB1, CB2 receptors, meaning humans that lived 300-400 years ago, are their brains receptive the same way that current humans are? Because in a way, my opinion is, or my question is, basically with the potency of cannabis or with the way humans consumed cannabis in the past, are there any similarities to why we have CB1 and 2 receptors in our brain now?
Right.
Meaning that the choices humans made in the past, like, several hundred years ago, is that the reason why we have receptors in our brain currently? Because
Right.
at this is like, I don't know if this is like evolution put those receptors there,
Right,
or is it,
right.
am I asking the wrong
You're
question?
you're asking a very difficult one. It's kind of like some of the why questions are always very difficult. Is there something changing over evolution over time? There's no doubt that there's things that are changing over time, and I could apply that to everything, not just the cannabinoid system. I think the unfortunate thing is that we've had very little amount of money to be able to study cannabinoids. And so, I'd love to be able to go back and say, if we did like we did with opiates, could we know a lot more today about cannabinoids like we do with opiates? And yet we still make the mistake with opiates. I mean, so there's no doubt that there's changes. Why are they there? I don't know. I love to think about the idea of why does the human brain have so many CB1 receptors? And I don't think it's right to say that, they're there to get high or to have psychotropic. They're there for some reason. I do think that they're regulating synaptic connectivity. And so hence some of our studies, our long-term studies, are to determine, will it affect cognition? Now, there are a number of papers out there that have shown that, but I think that we have to continue to sort of challenge ourselves to say, does it affect it? How does it affect it? Does it improve it, or does it, impair it? And is it different in different areas? And so I think there's a lot of things that we still need to do to determine why they're there, what are their role, what are their function, because we know that when you're during development in neonate, you have a ton of neurons making a gazillion connections. And actually what happens during the growth of the nervous system is there's dieback of neurons. So you have what we call pruning. We have nerves that actually die back so that you get a strong, good connection. And so sometimes that interaction of losing neurons is actually a learning process. As well as we know that when you also learn, you also make more synaptic connections. And so it goes both ways, where sometimes you make more connections as we learn, and then in some cases when you're learning, you're actually losing connections. And so understanding how cannabinoids play a role in that is very important, because we know for a fact that the receptors are there.